free to like unmute yourself. Okay, so my name is Aya, and I'll be talking about protein design using interpretable models. So first, let's us think about this world where drug discovery is a very, very simple task. It's so simple as if you are just calling an agent and you give that agent a sequence and you just ask it to do perform some sort of task that would usually takes months by a wet lab scientist. So for example, you want to decrease the hydrophobicity of a sequence with a certain constraint, like a maximum edit distance. And then that agent would just go call some model, and then they would basically return a sequence that has the exact things you asked for, a sequence with a decreased or increased property with some sort of constraint applied. Maybe you would even want something more. Maybe you would want to change, you have a bacteria and you would want to change this sequence to human, and then they would be able to do that as well. Or maybe you want to have an even harder task like a de novo design where you have an antibody, but you don't have a seed for it, and you ask the agent to basically design you the seed. So if we want to make this as like quick as possible and basically have an answer in a few seconds, what do we need to do? Well, we first need to be able to have a model that understands English. So, for example, when I tell it I want to I have the sequence and I want to decrease the hydrophobicity by 5 edits, it knows that the task is decrease the hydrophobicity and the constraint is 5 edits. The second thing is that we need the model, the model that this agent calls to be able to understand proteins. But not only understand like what is a protein, but understand the different concepts in a protein and understand how to change them and generate new protein in a reliable manner. So for the first task, it's good that we already figured that out. And there's many models and agents out there that do a great job, such as Gemini, Claude, ChatGPT, DeepSeek, and so on. For the second task, there are protein language models that, such as the ESM series or even the Lobster series that is developed by Prescient, and many more. But the thing is, with these protein language models, they understand how to represent a protein, but what they do not understand is the biophysical properties, biophysical concepts of a protein, and how to change these concepts to make a new protein. So that's where they lack. And this is kind of what we will be trying to fix in this work. So I'm going to start by giving or like an overview about masked language models, what they are and how they're trained. Well, masked language models were introduced in 2019 in a paper called BERT. And basically, the high level idea was that we are going to train a model in the to complete a sentence. And we know humans have been doing this for a while. We have took exams when we were kids about this. So they thought, okay, let's train a model exactly the same way, where we have a sentence and we're going to mask out a few tokens and ask the model to complete it. And then we're going to add a loss on top of that to see if it completed it right or wrong based on the original sentence. So when people thought about this task, they did really well. So they thought, okay, let's translate it to proteins. And what they did is exactly the same way. They took a protein sequence, they masked some amino acid, and then they asked the model to predict it. And this is the types of model that is like super famous for protein language model, such as ESM, BERT, Lobster, and many, many more. Most of them are this masked language model protein. So people then started to think about, okay, conditional mass language model, which is mainly done for conditional generation. So how do we change the mass language model to a conditional mass language model? And for English, basically what they said is like, okay, we can add a tag at the beginning. So for example, I want to generate a female, then my tag, I want to generate something that has the concept female. So I'll add a concept female as a tag so that the sentence, the model learns to complete, I am a strong woman rather than I am a strong man. So when people started working on protein, they thought, okay, we're going to do exactly the same thing for protein. So we're going to add a tag such as it's the species is bacteria, so that when the model is generating, it learns, it knows that, well, we want to generate a protein sequence of a bacteria. Okay, but these models have some problems. So for example, these models are basically considered of an encoder-decoder architecture, but we do not know what this encoder learned at all. And for the decoder, we do not know how they are making the decisions. We also do not know what the decoder can or cannot do. So we don't know what the model really is capable of. Like, for example, I might ask it something and it does not know how to do it, and I'm not sure if this is the case or not. And finally, we don't know how to debug this model or how to fix the errors it's producing. So these are the main problems of like black box mask language model. And so even more so, if we can think about that a model, a conditional a mass conditional language model can easily like ignore the tag. So for example, if I tell it species and I give it a protein sequence, it can completely ignore the species because it did not need the species for generation. So what people started to do is saying, well, okay, we're going to give it the species. So for it not to ignore it, we're going to add like a classifier head or make it predict the species as when it's done generation. But one can think that there can be basically a residual connection between the input and the output, where it's like, okay, you gave me the species as an input, I'm going to predict it as an output, and I'm not going to be use this information to learn anything biologically meaningful about that protein. So these are the main problems with like the types of conditional language models. And in this talk, I'll be talking about concept bottleneck mass language model, which is a new type of models that tries to address every single problem that we discussed about. Okay, so back to the black box mass language model. So first we said that we do not know what concepts the model have learned. And so let's try to fix that. So we will start by having the sequence as is, and we will have the encoder as is. So the encoder in the model on the right is exactly the same as the encoder on the model on the left. But in the concept bottleneck model, what we're going to do is we're going to take the first token, which is the CLS token, and we're going to pass it through a concept bottleneck module. And that module, we're going to predict the concepts that were present in this amino acid and in this protein, sorry. And the idea is that why do we do this at the end of the encoding process, because due to the cross attention over and over in the encoder layers, it had already seen all the sequence. So by the time it's done encoding, like the first token, which is the CLS token, had already seen all the other tokens. So we can just take it and predict the concepts that were in it. Okay, now that we have these concepts, we want to generate new tokens that do not have this information in it. So what we do is we pass the old tokens in a network called an orthogonality network. And basically this orthogonality network just gives us new tokens that do not have information about the concepts in them. And we call these tokens the unknown part of a model. And basically the concepts that we have, we call known. So we add it back together. So you can think about this step as a decomposition step where we took an embedding that had concepts and things that are not concepts. And first we extracted the concepts from it. Then we basically tried to find an embedding, a representation of that embedding that does not have these concepts. And then we put them on top of each other as known and unknown. And then we basically have a linear layer on top and use this linear layer to make the prediction. So this concept bottleneck basically tells us make sure that we learn the things we care about, and it tells us that make sure that we understand the protein, how the protein are made, and make sure that we learn the things that are important to the biophysical properties of the protein itself. So for the model, if we want to know how the model is making the decision, we couldn't do that for the one on the left, but for the one on the right, basically we can look at the linear layers and see it depends on which concept. Did it depend on the known or the unknown, and how much did it depend on each? And then we know exactly what the model is making, how the model is making the decision. And basically, we know what the model can or cannot do by basically looking at the weights. So for example, if the model did not learn a certain property, then it won't be able to control for that property. So we know, like, regardless of what the input is, if the model waits for that concept is like almost zero, it won't be able to know to control for it. And we basically, yeah, so that's the importance of the linear layer. And we know also, like, how much exactly did the model depend on every concept by multiplying the activation of that the weights with the activation of the concept to know how much the model depend on the output depend on that concept. So basically this kind of like solved all the problems that were on the model in the left. So yes.
The tire. Some question about your proposed architecture. I'm seeing the model from the transformer encoder to the next layer, and then to the orthogonality network. I feel personally, I think I've missed what exactly happens to the sequences and to the model, how it is learning. But also, when you look at the unknown and the known layers, I feel I still need more, like, what really happens underneath those two layers, those layers from orthogonality upwards. But also I'm seeing the intermediate result after the transformer encoder. The next layer is connecting to this CBM layer. And I'm also wondering what is happening within that layer. So the result from there goes into the known layer. So I just need a little bit of light within the subsequent layers after the transformer encoder.
So yes, so the transformer encoder, what it does is that it gives us an embedding for every single token, right? So let's say that your sequence is CLS MKQ, and then there's a mask TPIT, end of sentence, right? This is the input to the transformer encoder. The output is an embedding for every single token.
Okay.
So that's the output of the transformer encoding. So you're going to basically take the CLS token, the output, but you know how the but at when we're done encoding. So when we're done encoding, basically all tokens have seen every other token because of the cross attention, right? So the CLS token has saw the whole sequence of the amino acid at this point. So we take this, so it has information about the whole protein. So we take this sequence and we pass it into a model that we train. This whole network is trained end to end that predicts the concepts we care about. So let's say that we care about affinity, hydrophobicity, we care about species. So we're going to take that CLS token and create a multitask classifier that is going to give us values for every single concept we care about. And that's exactly like what this concept bottleneck module does. So these are just a model that takes an embedding, which here we use the CLS, and gives us a value for every single concept in that model. And then what we do is, okay, so now we have the tokens from the token from every single tokens from the entire sequence, and we have concepts in the entire sequence. So we want to create new embeddings for the tokens, but we want these embeddings to have a property that they don't have this concept in them. So one way to do that is to pass the embeddings into a network, and a different network, which is just an orthogonality network. And you can think about it as a network just that tries to find like a disentangle, that tries to disentangle two embeddings. So we give it all the tokens, and then it gives us new representation of the tokens that does not have the concepts in them. And I'll get to how the losses we enforce on this different layers later in the talk, but that's kind of like the high level idea. So now these tokens for all the sequence, they do not have the information about the concepts. So that makes our unknown part. And then we put the output, we concatenate the output of the concept bottleneck, which is the known part, on top of that. So now the embedding is the known and the unknown part, which has the same exact embeddings as the blue one before, but it's just disentangled, so that we know, like we kind of like localize the information of the different concepts in different areas of the embedding. And on top of that, we put a linear layer, which is so that we are able to kind of know the weights of the different concepts and the weights of for every single token. So this is kind of what is happening here. Does this make sense?
Yeah, absolutely. Uh, yeah, it makes more sense.
Okay, great.
Mm.
Um, okay, so now when you want to make the prediction, you basically, so let's say we want to make the prediction for E, we basically look
Maybe,
at,
maybe another question would be about the orthogonality network. Like, would you say underneath, didn't know whether this is in the paper, within the orthogonality network, it's you completely reinvented sort of a new algorithm underneath there,
no,
or
it's
it is
just a
something that
feed-forward
you
network.
just,
You can think about it as just a feed-forward network. Is this a
oh,
projection
okay.
to an embedding? Yes, but
Okay.
we'll get to how we train it. But it's just a
Feed
feed-forward
forward.
network. It's nothing fancy.
All right. Okay, thank you.
Okay. So, okay, now when you want to predict E, basically what you look at is you look at the known and the unknown part for that token, and then you make the prediction using a linear layer, and that's kind of like how that happens. Okay. And yes, and for debugging, you can basically, during training, you can look at the different losses that we add, and you can look at the weights to make sure that it actually learned the concept that we want. So this is how you would debug this model. Okay, so this is what the model looks like. So in a little bit more details, so we have the mask sequence, we have the encoder, and then we have the hidden token representation, which is H. We take the first token, hidden token representation, which is H0, we pass it through the concept bottleneck network, and then we pass all the tokens to a feed-forward network that gives us new tokens, which is, we call it the unknown, and we put back the output of the concept bottleneck, which is the known, and then we make the new prediction of X hat. So how do we train this model? First, we just have the regular cross entropy loss for the mask token. So basically predict that a loss that makes sure that we predict the mask to be the token E. And this is like regularly what you would do for mask language modeling. But now let's look into this network that I called the concept bottleneck, the module that I called concept bottleneck module. So what does it do? It takes H0, which is the CLS token, and it makes a prediction for every single concept here. So every color represents a different concept. But we also have an embedding that represents the representation of the concept. So we have, and this embedding is a learn embedding. So we have a value for the concept and an embedding that represents the concept. And what we do is we multiply the value with the embedding, and we basically concatenate all of these for all of the concepts that we care about, and this makes up our known. And on top of the concept, the value for the concept, we add a concept loss to make sure that it aligns with the ground truth concept. So, so for example, if we want to make sure that this sequence is a bacteria and the species is correct, we would add a concept loss to make sure that the prediction is a bacteria, and then we would multiply that prediction with the embedding that represents the species, and that is going to be part of our known embedding. And we do this for everything else. Okay, so now back to the orthogonality loss, the orthogonality network that you asked about previously. So the idea is that you want to get a representation that has that does not have information about this known part. So what one simple way to do that is just to minimize the cosine similarity between the hidden representation that is outputted from this network and the known representation. So by minimizing these two the hidden representation and the unknown and the known, you basically kind of create an embedding, which is the unknown embedding that does not have information about the known. And so our total loss is just the mask language model loss, the concept loss, and the orthogonality.
Dr. Haya,
Yes.
I'm quite curious, and it's because the work is getting exciting. On the previous slide, when you're showing the concept bottleneck underneath it, yes. When we think about a protein, we are looking at these sequences and chains of amino acid groups, things like that. And I'm not a biologist, but I'm using the basic information that I know. And looking at the concept bottleneck module, this level of sort of say classification of the different sort of groups or chains within the protein, and those that we know maybe the functions within the protein, and those that we don't know. Do we perform this classification at either the smallest unit of the protein and whatever that could be, or being at the groups that are forming up the protein sequence itself? So at which level do we start this sort of classification with
So,
the
okay.
corresponding groups, also the functions?
So, okay, so here what you give the entire sequence and you have one CLS. So the properties that you are going to have in this concept bottleneck is going to be on the entire sequence. If you are if you want something that is like on a very short like a subset of properties, then yes, then you just give it a very short sequence. But like right now we just assume that we are adding properties for the whole protein, not a subset. But this is a very like a very good question actually, because like sometimes properties change from one part of amino acid to another, and the sequence have like yeah like a very long sequence, and it can have different properties. But here we're just looking at the global properties. So you can think about this whole protein comes from a bacteria, um, or this whole protein has affinity this value to this target, for example, rather than okay, amino acid from 5 to 10, what happens there?
Oh,
um,
okay.
does
Okay.
that answer?
Thank you. Yeah, sure.
Okay. Great. So we train this model end to end from scratch using this like these three losses. So the mass language model loss, the concept loss, and the orthogonality loss. And basically, okay, so how do we intervene on this model? And by intervention, it means that how do we want to change a property and see the effect on the output. So we do this by basically first to do this, we need to know which tokens we need to mask. So one simple way is to just randomly mask a token and then intervene on a concept. But the problem with that is that like you can let's say that you want to decrease hydrophobicity, and then you randomly mask a token and then say okay, decrease hydrophobicity, but you ended up to randomly mask tokens that have already very low hydrophobicity. So changing these tokens won't decrease hydrophobicity. So random masking is not the right idea to go. The other option is doing occlusion. And what occlusion is that, okay, we're going to perturb every token in the sequence and see which one affects hydrophobicity the most. The one that affects hydrophobicity the most, we're going to mask that out and then predict it. But the problem with this is it's super, super slow, because if the protein is very large and you want to see which the effect of every single one, then yeah, that's slow. So the best thing you could do is do something like gradient times the best way we could find is basically get the gradient of like the input with respect to the concept, and then multiply that by the difference between the input and the regular and the mask. And you can think about this as a single step integrated gradient, basically. That this What does this equation tell us? It tells us, okay, if I change this, how much did this protein contribute to this concept with reference in comparison to a reference point. And here the very obvious reference point is the mass token. So how much did this protein contribute to this concept in comparison to like the mask. And then you multiply that, and then that basically tells you which amino acid was most important for that concept, and then you can mask it to increase and then increase and decrease the properties. So yeah. So any questions before I move on? Because I'll start like talking about experiments now.
Um, I'm still I'm still thinking now. When you say like predicting a certain sort of property, but uh, we are now when you go back to the previous slide, you've mentioned the property like hydrophobicity. But then you talked about the previous, I think the second method, which was occlusion, and you said it is slow. And for me, it sounds like occlusion would give the the most sort of like accurate results because it sort of like tests out every part
Yes.
of the
So
protein.
so this does exactly the same, but it does it in one step. So basically,
Okay.
yeah, so occlusion, you would have to perturb every single part of the protein. And this, you just get the gradient of the concept with respect to the input and multiply by that by the change if it was a mask. So this in one step, you get the contribution of every single sequence, every single token in the sequence. Rather than doing it one token at a time, you get it all at once. And then you can know the contribution of the most important one. So this is something that is an approximation to occlusion, um, but it is much faster.
Okay. Okay, just to end or to sort of abstraction, when it's I get it now when you have the input, so that means we have occluded sort of like the mask. But what is the gradient? Like we know, okay, the gradient when we think about it in terms of computation, but in this concept, in this computation, is it the normal gradient as we know it?
Yes, so it's the gradient of so usually like how people know the contribution of the input with respect to the output, they take the gradient of the input, which is
Okay,
here it's
okay.
the token, with respect to the output, which here is the concept. So this tells us the contribution of every single input to the output. And this is kind of
Oh,
like a
great.
way that people measure the contribution of every token of inputs to the output by taking the gradient. But like here, it's because we want to see the effect of changing the input. So we compare that contribution from the input to the respect to the output with like just a mask when it was like a reference, which is like a reference point. And and then when we multiply the gradient value with the difference in the embedding itself, this gives us basically a single sub integrated gradients. This is like a method, a really famous method, and this tells us the contributions of every token in a sense.
Perfect. Um, that's great.
Okay. Okay. So basically, yeah. So you can basically think about like this model that you can start with a protein, and then you can think about having a knob that changes the different concepts and generate proteins with different properties based on that. Okay, so let's talk about first about like scale. Can you scale this model? So we trained a 24 million parameter model, a 150 million parameter model, a 650, and a 3 billion. And we train it on UniRef 50 and Swissport with over 700 concepts from some of them are BioPython concepts and other are like Swissport labels. So when we look at like the quality, the perplexity, when we compare the perplexity of our model with like other models. So the model here is like this concept bottleneck. The last one, the concept bottleneck protein language model, you can see that it is comparable to like the ESM2 series that don't have concepts in them. So we can scale. This is one thing, and we do not lose quality. So, okay, how do we use this model for control, interpretability, and debugging? So first for control. So just so that to have like in silico experiments, what we're going to do is we're going to take a set of proteins and we're going to calculate a set of BioPython properties, such as, for example, gravy. And then the second thing we're going to do, we're going to intervene on a concept, either by increasing it, which is we call it a positive intervention, or decreasing it, which is a negative intervention. And then the model is going to produce as a new sequence. And then we're going to take this new sequence and then pass it through BioPython again and measure the value of this property. So these are kind of like the results we have. So the first model is a regular conditional model that we talked about at the beginning, where you put the tag at the beginning or the values of the concept. The second one is a classifier, a concept classifier model, which is basically similar, very similar to the one ESM has, which is that you put the concept and you put you put the concept at the beginning, but you also have a classifier head and on top of that, and then you use both to make the prediction. The third one is our model, where you basically predict the concept from the internals of the model, and then you use that prediction to make the output. And you can see that like with for the 24 million parameter series and for the 150 million parameters, we significantly outperform the other models in terms of control. Okay, and we can look at also the magnitude of change. So you can see like how much the property changed. And what we find is also in the concept bottleneck protein language model, you can see a very, very big change in the property because we know where to mask, we know what to mask. So when we mask it and we change the value, we get the right thing basically. And yeah, so that's in terms of control. If we look at the shift in the distribution, so here we had the original distribution and we shifted a property positive and negative. So the original distribution will be the dotted line here. And then you would want the positive to go to the right and negative to go to the left. So here, concept bottleneck is the red model. So when we shifted it to the right, to when we did positive intervention, you can see that the colors for the properties for the model that is red went in the right direction, and when it was negative, it went also in the right direction, while the other models, which is like conditional model and the conditional model with the classifier, it didn't actually go in the right direction at all. So we can see that we are able to shift the distribution of a lot of samples. And another cool experiment that we tried is we wanted to see if we can continuously shift the distribution iteratively. So we basically fed the model the input again and see if we can change the property. And we could see like here, for example, that every single iteration, the property kind of went up. And here in every single iteration, the property went down because this was a negative intervention. That's what we would want. And so it's good to see that we can continuously increase the property. And we also looked into increasing two properties at once. So here we changed, I think this was pH 7 and gravy, and you would basically, the parafrontier would.
Doctor, I'm just curious on the previous slide.
Yes,
Um,
there's an
sorry.
echo, I think.
Yes, no, this one, yes, that one. Um, Positive intervention, negative intervention. When I look at the diagram below, positive intervention, well, I'm seeing like some sort of perturbations moving upwards and downs. But the question is, what is this positive intervention and negative intervention? It is like, does it happen during training?
No,
And
so this is intervening. Well, we take the sequence, we change the concept values like I described with like you basically mask the tokens that are important and then you change the value in the concept bottleneck. So
okay,
we go back to this model here. The okay, you change the value here basically. You intervene in
okay,
the model to change the values of the concept.
sure, makes sense, makes sense. So what would a negative intervention look like if I change the value like,
So, for example, let's say that a negative intervention would be you have a property and you want to decrease it. So you're decreasing the
okay,
property. So let's say that this was one and you make it zero. This would be a negative intervention. A positive
sure,
intervention would that this was zero and you make it one.
okay, thank
So
you.
a negative intervention decreases the property. A positive intervention increases the property. And if we go back to here, you could see like in the positive intervention, this is the property value or the change in property value. So you would want it to, if I intervene positively, I'm increasing the property value. So I want the distribution to shift to the right, right?
Okay,
And this is what happens in the red distribution. It shifts to the right.
I see.
If it's a negative intervention, I would want the distribution to shift to the left. And this is what happens here too.
Okay. So in terms of the results, like where do you find that you find yourself with an optimal sort of like intervention for the best result?
So, I mean, it depends. You want to change, you can change it as much as you want, but you want to basically have some sort of constraint where you say, I want to change a property without changing the protein too much. So, for example, you would say, okay, I'm going to only mask five amino acids, and I'm going to find that amino acids that I'm going to mask using like this equation here that we had. And once I find them, I'm going to mask them, change the value of the concept in the concept bottleneck model, and then get the protein, the output. And maybe you would want something more aggressive. You'd say, okay, I change 100 amino acids. So yeah, that's up to you basically. Like how much
Okay,
you that like how much you want to how much different you want the amino acid to be.
sure.
And
Makes
this slide
sense.
here is just it's the same thing. So here, so okay, so and here this is sorry, this I should have said this. This is naturalness. So how natural the protein is. So basically the goal is to shift to the right and the left without changing this y-axis, because this tells us how natural a protein is. And you can see that like our model shifts the distribution right and left, but it does not affect the naturalness that much. And that's kind of what you want.
Okay, sure.
Yeah, and here this is like just multi-property. So here we're changing two things at once. You can see that like also the model here, we're increasing both. So we would want sequences on the top right, and here you would want it on the bottom left, and you kind of see that, yeah, we were able to get that. So this is in terms of control, where you change the concept and you get a protein that has the new property that you want. Okay, in terms of interpretability, well, what does this offer? Well, it offers us local interpretability because we can basically, for a particular sequence, we know we can look at the output of the concept bottleneck module and know exactly what the concept of the model relied on, what are the values of the concept for a particular protein. And that is just by looking at the output of this bottleneck module. And the other things is that we also can have like global we have like global information, which basically tells us the insights about the relationship between the different concepts. And we do that by looking at the weights of this linear layer. So for example, this is from our train model. These are the weights for pH6 and pH7. And these are the weights learned by the model. And if we take a closer look at these weights, we find that they have a very similar pattern, except for the like these two amino acids. I think it's D and E. Oh yes, so these two have like negative. These two have negative weights for amino acid D and E, which is exactly how both pH 6 and 7 have, and amino acid R and K have positive pH, positive value weights for pH 6 and 7, and they only differ in amino acid H, which is exactly like how biology is. So basically, it is more positive when it is in a lower pH and negative when it is a higher pH. That's what the model learned. So this kind of like allows us to, if we did not know this, now we know what the model learned. If we already knew this, we validate that the model is actually learning stuff that humans understand, like that humans know. So it verifies that the model is like doing the right thing. And okay, so the last thing is like debugging. So what model debugging means, it means that first we want to be able to detect and errors during training and during testing and fix them. So basically, the concept bottleneck model allows us to be able to have this interpretable, human understandable concepts that we can basically ask questions is like, did the model learn the concept of our interest? Is the model relying on spurious concept? Is what situation will this model fail? And are there any unwanted correlations? And basically, we can answer all of these questions by just simply looking at the weights, the validation losses for the different concepts. So for example, if the model cannot predict a concept, then it did not learn this concept. So we can verify that it learned a concept or not by just looking at the validation loss for a different concept. Also, we can look at the weights of the linear layer to tell us which concept the model relied on, and we can look at that for, we can also use it for intervention to test the effect of changing the concepts on the output. So this is an example of like two models. One was a good model on a particular concept. The other, I corrupted it and trained it. And if we look at the weights, we can say that the model on the right basically has weights for different amino acid for that concept, which means that it actually learning something about amino acids for that particular concept, but the model on the left did not. So without me having to basically test this model in a wet lab, I can just look at the weights and say, okay, this model will not work for aromaticity. And that means that, yeah, I should not be using this model for this property because it did not learn it. And if we did actually try to change this, we can see that the bad model, was not able to change at all, but the good model, which is the red model, was able to change that particular property. So this is kind of like how you can use the weights to debug the model. So yeah, so I basically talked about like these different things that you can do with this model: control, interpretability, and debugging. I just want to say that this work was done with like my amazing collaborators, all of which are here. And yeah, if anybody like have any questions, anybody has questions, I'm happy to go over it again.
Thank you, Dr. Aya. This has been revolutionary and exciting work. And I think every day when I look at the protein like focused research with LLMs, it keeps on, I think, growing and growing. And for us, our interest is to find a footing at least and find ourselves at also at the forefront of this research. Thank you very much. I think for me, yeah, all the gray areas have been cleared. I don't know whether we have questions from other members. And so many members have been sending me messages that they are moving up and down. Tomorrow is a public holiday here, and oh yeah, Dr. Aya also. I'm I'm assuming she's a Muslim, and yeah. So tomorrow is Eid. Allow me wish
Yes.
you a
Oh,
nice
thank
Eid.
you. Thank you so much. Yes.
Yeah.
Yes. Yes, thank you so much.
Yes. So I think members don't have questions today,
Okay, if you
and
guys have any questions, feel free to email me anytime.
sure. And I want to thank the members for turning up, but also to encourage you members to push when it comes to this research. You never know where this work is going to participating in these reading groups, where it is going to take us. Probably next time Dr. Aya will pop up and is looking for students. Who knows? So I would encourage you that you push the boundaries and read these papers, and maybe you can come up with a problem. I already see like some possible extensions of Dr. Aya's work. Maybe I could be wrong, but at least the feeling I've gotten. So, but still, thank you very much for turning up, and special thanks to you, Dr. Aya, for really at the end
Thank
of
you
the
so
day
much.
making it through. Yes,
Thank you. Have a great day. Thank you.
yes.
Happy Eid.
Happy Eid too, and goodbye. Okay.